Greetings of the day to the viewers. Myself, KL Mary, working as Assistant Professor in the Department of Pharmaceutical Analysis in the Nirmala College of Pharmacy. HPLC is the high performance liquid chromatography or high pressure liquid chromatography. Here we will use the high pressure in order to resolve the components. Coming to the objectives of the HPLC, it is used to analyze the mixture of the components and to perform the stability studies and to analyze the components present in the urine or the blood samples of the drugs of abuse. Coming to the principle of separation in the HPLC was adsorption. The separation of the components will occur in between the mobile phase eluent and the stationary phase adsorbent. The compounds which are having the more affinity towards the stationary phase will elute slower at the later. The compounds which are less affinity towards the stationary phase will elute faster. In most of the analysis in the laboratories, we will do the drug substances and the pharmaceuticals having the polar in nature. So we will use the non-polar stationary phase, that is the reverse phase chromatography. This is the HPLC equipment we have in our laboratory. Cyber Labs is the manufacturer of this HPLC equipment. Coming to the components of the HPLC, first one, mobile phase reservoir. In the mobile phase reservoir, we are using the HPLC grade solvents. Mixture of the solvents are the single solvent is used as the mobile phase reservoir. Based upon the nature of the chromatography and the nature of the stationary phase used and the nature of the components to be separated. Before using the solvents as a mobile phase, it should be degassed. The degassing will be done in the ultrasonicator or in the vacuum filtration pumps. In the vacuum filtration pumps, why you are degassing the mobile phase used in the HPLC in order to remove the dissolved gases present in the mobile phase. Second component of the HPLC was pumps. These are the pumps in the HPLC equipmentation. It delivers the mobile phase into the column. The pumps can withstand the high pressure of the 6000 to 9000 pascals, and the pumps is having the two types, isocratic and gradient mode. In our laboratory Cyber Labs model, we have the isocratic pump. It can deliver the mobile phase only in the constant ratios. In the gradient pumps, it will deliver the mobile phase in the variable ratios. Generally, the flow rate of the mobile phase based upon the mode of chromatography, that is the analytical mode of chromatography or the preparative mode of chromatography. Generally, you can use the 1 to 2 ml of the flow rate of the mobile phase into the pumps. Third component of the HPLC was the sample injection system. Here we are having the manual injection system in which the sample was injected through the Hamilton syringe. This is the Hamilton syringe. Through this Hamilton syringe, you can inject the sample into the septum of the injector. The capacity or the volume of the sample injection was 20 microliters. Excess of the sample will comes into the collector unit. After loading the sample into the injector system, we can inject or trigger the sample system into the inject mode. Then the sample in the injector will goes along with the mobile phase, it will goes to the column. The fourth component of the HPLC was the column. Column is the heart of the HPLC, where the separation of the components will occur. Proper selection of the column, that is chemical nature and dimensions of the column, will affect the resolution and speed of analysis and the peak symmetry. In our lab, we are having the intrinsil HPLC make and the ODS column, that is octadicyl silane, and the length of the column was the 150 millimeters and the internal diameter of the column was 4.6 millimeters. The stationary phase particles filled inside the column of the 5 micron size. After the separation of the components in the column will goes to the detector. The detector will analyze the components and give signals to the recorder. Recorder is connected to the PC. In our labs, we are having the UV detector. The analysis is based upon the absorbance of the separated components. In advanced equipments, there is a different type of detectors, that is a fluorescent detectors and the refractive index detectors and photodiode array detectors. Before analyzing the sample, you can check the calibration status of the equipment. HPLC equipment was calibrated once in a 6 months. The calibration of the equipment was done with the caffeine solution. Why? Because the caffeine solution was used. It is a non-volatile substance and the readily available. It has a strong absorption in the UV region. Various parameters was calibrated in the equipment. Coming to the pumps, the flow rate consistency test was done and the flow rate accuracy test was done. Coming to the injector precision test and injector carryover test and the injector volume accuracy test will be done. Coming to the detector, we will check the linearity of the detector and wavelength accuracy and the baseline noise. By checking all these parameters, you can confirm that the equipment was calibrated. Then you go for the analysis of the sample. Before analysis of the sample, you can wash the columns and the washing of the pumps was done. Washing of the pumps is termed as the purging of the pumps in order to remove the previously analyzed particles are present in the pumps. It should be purged and the column will be saturated with the mobile phase what we are using in the sample analysis, that is called as a washing of the columns. Coming to the analysis of the sample, today we are observing the analysis of the paracetamol drug substances. For the estimation of the paracetamol by HPLC, you can use the methanol as a solvent and the water in the ratio of the 50:50. You can use the HPLC grade solvents. By mixing of the 50:50 mobile phase, the mobile phase was degassed by using the ultrasonicator. After that, the sample was dissolved in the mobile phase of the concentration of the 10 microgram per ml and the 20 microgram per ml. The mobile phase was filled into the mobile phase reservoir. Then coming to the pumps, you can enter the flow rate as the 1 ml per minute and the detector wavelength was 257 nanometers. The sample was injected into the injector port by using the Hamilton syringe. The solution was sucked into the Hamilton syringe. After sucking the solution into the Hamilton syringe, we observed that any air bubbles was entrapped in the Hamilton syringe. By confirming that without air bubbles are entrapping in the Hamilton syringe, we inject the sample into the injector port through the septum. The sample injection was in the load mode. Then we will observe the software in the PC. After observing the straight line as the base, then we will turn the sample injection system into the injector mode. Then the sample was injected into the column along with the mobile phase. In the column, the separation of the components will occur. The separated components will goes into the detector. The detector will analyze the components. The analyzed components will gives the signals to the recorder. In the recorder was connected to the PC. In the graph, we'll observe the separated components as the peak. The recorder will give response as a chromatogram. In a chromatogram, the x-axis measured was the retention time in minutes. Y-axis has peak area or intensity of absorbance. Retention time was the time between the point of injection to the appearance of the peak maxima. Each component of the analyte has different retention time values. Peak area was the calculated by the area under the curve. The concentration of sample is obtained from the area of the sample is divided to the area of the standard and multiplied with the concentration of the standard. Area is the proportion of the concentration of the component present in the analyte. Applications of HPLC being the sensitive and the versatile technique, HPLC used in the wide variety of the industries. Apart from the pharmaceutical industry, it is used in the chemical and the petrochemical industry. In the forensic applications, it is also in the environmental application. It is also used in the biochemical industry, and it also separation of the components in the biotechnology, and it is also used for the estimation of the compounds in these fields by the qualitative and quantitative analysis. In the qualitative analysis, comparing the detection times of the sample with the standard, we can analyze the identification of the compound. Coming to the quantitative analysis of the compound, it is done by the direct comparison method, calibration curve method, and the internal standard method. It is also used in the multi-component analysis. It is used for the detection of impurities in the sample, and it is also used for the determination of the purity of the compounds. I am very much thankful to the principal sir and management, HOD of the Pharmaceutical Analysis Department of the Nirmala College of Pharmacy for giving this chance. Thank you, one and all.